Ever wondered how DNA replicates? Today we delve into the fascinating world of DNA replication, specifically focusing on the replication fork formation. DNA replication, a critical process in all living organisms, is the basis for biological inheritance. Now, within this intricate process lies an interesting formation known as the replication fork. This site of active DNA synthesis is where the DNA helix unwinds and single strands of the DNA start to replicate. Picture it like a fork in the road, paving two paths for the creation of new DNA strands. Formed during DNA strand unwinding by the helicase enzyme, the replication fork exposes the origin of replication, setting the stage for the synthesis of new DNA strands. This is just the tip of the iceberg, however. The replication fork formation plays a much deeper role in DNA replication. Let's dive into the mechanics of the replication fork formation and its function. The formation of the replication fork is a crucial first step in DNA replication. This is where the magic of life's blueprint duplication begins. Let's dive into the details. The replication fork is formed during the unwinding of the DNA strand, a process facilitated by a star player in our biological drama, the helicase enzyme. Picture helicase as a zip runner separating the two strands of DNA, revealing the origin of replication. This origin serves as the starting point for the replication process. Once the DNA strand is unzipped, we meet another critical character in our story, the primase enzyme. Primase jumps into action, synthesizing a short RNA primer. This primer is crucial because it provides a starting point for the next phase of replication. Enter DNA polymerase, the builder in our process. This enzyme attaches itself to the RNA primer and begins the elongation process. It adds nucleotides, the building blocks of DNA, to the RNA primer, extending the strand. Now, let's talk about the direction of the replication fork movement. It moves in the direction of new strand synthesis. And here's an interesting fact. The new DNA strands are synthesized in two orientations. One is the 3' to 5' direction, known as the leading strand. The other is the 5' to 3' orientation, known as the lagging strand. The replication process on the leading and lagging strands occurs in two opposite directions from the replication fork. This is why we say that the replication fork is bidirectional. So we've formed our replication fork, introduced our key enzymes, and started the process of DNA strand elongation. The stage is set, the players are in place, and the drama of DNA replication is well underway. So the replication fork is formed and ready to move, but which direction does it go? Stay tuned as we delve deeper into the direction of the replication fork and the synthesis of new DNA strands in our next scene. The replication fork moves in the direction of new strand synthesis. But here's the trick. The new DNA strands are synthesized in two different orientations. Let's imagine the replication fork as a Y-shaped structure. At the Y intersection, there are two paths, one leading to the 3 end and the other to the 5 end. These paths represent the two new DNA strands. The one moving in the 3 to 5 direction is the leading strand, while the one in the 5 to 3 orientation is the lagging strand. Now, picture the leading strand. It's the easygoing one, synthesized continuously by the DNA polymerase enzyme. This strand begins its journey when the DNA strand unzips and separates, creating a short piece of RNA, known as a primer. This primer acts like a starter's pistol, signaling the DNA polymerase enzyme to start the synthesis of the new strand, the leading strand. This process occurs in one smooth, continuous motion. On the other hand, the lagging strand is a bit more complex. It's synthesized in a discontinuous manner. As the leading strand synthesis occurs, it exposes small short strands or templates. These templates are then used for the synthesis of what we call Okazaki fragments. These fragments are added to the strand between the primers by the activity of the DNA polymerase. This process is a bit like building a brick wall. where each Okazaki fragment is a brick placed precisely in the correct spot. What's fascinating is that these two strands, the leading and lagging strands, are replicated in two opposite directions from the replication fork. This makes the replication fork bi-directional. One might say it's like a busy two-way street with traffic flowing smoothly in both directions. The mechanics of replication fork and strand synthesis is a meticulously orchestrated process, isn't it? But it doesn't stop here. The leading and lagging strands of DNA have distinct roles and synthesis processes during replication. Let's start with the leading strand. This is the new DNA strand that's synthesized continuously by the DNA polymerase enzyme. Think of it as the simplest strand synthesized during the replication process. After the DNA strand has unzipped and separated, a short piece of RNA known as a primer is generated by the DNA primase enzyme. This primer binds to the 3' end or the start of the strand, thus kicking off the synthesis of the new strand, which we call the leading strand. The synthesis of the leading strand is a continuous process, like a smooth highway being laid down. Now, let's turn our attention to the lagging strand. This is the template strand that's synthesized in a discontinuous manner by RNA primers. As the leading strand is synthesized, it exposes small, short strands or templates that are then used for the synthesis of the Okazaki fragments. Named after the scientist who discovered them, these fragments synthesize the lagging strand by the activity of DNA polymerase, which adds pieces of DNA, the Okazaki fragments, to the strand between the primers. The formation of the lagging strand is a discontinuous process, more like a cobblestone road being built piece by piece. The newly formed strand, the lagging strand, is the result of the fragmentation of short DNA strands. So, in essence, we have two strands being synthesized in two different ways. The leading strand synthesized continuously and the lagging strand synthesized discontinuously. This all happens at the replication fork, where these two strands, the leading and lagging, are replicated in two opposite directions. And that's how the replication fork and the two DNA strands work together to ensure accurate DNA replication. It's a complex dance of molecular interactions that keeps life going. DNA replication is a complex yet beautifully orchestrated process. The replication fork, with its bi-directional nature, plays a crucial role in this. It ensures the successful synthesis of leading and lagging strands with precision and continuity. Next time you look in the mirror, remember, the replication fork is hard at work in your cells, ensuring your DNA is accurately copied. Until next time, keep wondering, keep learning.